Okay, here is my student presentation on Bertram Jacobs by Marshall Donaldson. To start off, a little background for his education. Professor Jacobs got his PhD in biochemistry, University of California, Berkeley in 1981. Got his BS in biology, Rutgers University, 1974. Here are some awards that he received. Arizona Governor's Celebration of Innovation, Innovator of the Year Academia 2006, nominated for Arizona Bioindustry Association Bioscience Researcher of the Year June 2007, Southwest Center for HIV/AIDS Humanitarian Award 2009, Distinguished Nominee ASU Parent Association Professor of the Year 2011. I'm pretty sure some of these awards were made up just for him, so must be a pretty good guy. For professional experience outside of being an ASU professor, he was also on the editorial board of Virology 2005 to present. He was on the Support for International Change 2005 to present, Center for Infectious Diseases and Vaccinology Biodesign Institute member 2006 to 2010, University of Biosafety Committee Chair 2005 to present. Those are just a couple that I chose. Research interest. Professor Jacobs is focusing his research on developing a vaccine for HIV. He's also interested in microbiology, molecular biology, and viruses. He's actually given a ASU kind of TED talk about the benefits of virus. It's pretty cool. It talks about how viruses are kind of like hackers, which is generally a bad thing, but if they're working for you, then it can be a good thing. So for this assignment, we chose to do the mouse hepatitis coronavirus A59 nucleoapsid protein is a type 1 interferon against an interferon antagonist. Whew. Pretty interesting experiment that he did here. There's the link if you want to check it out. The question, pretty much put simply, how do coronavirus affect the type 1 interferon system or the immune system? Hypothesis, if mouse hepatitis virus or MHV A59, which is a type of coronavirus, which is actually the reason I chose this experiment, because it was a coronavirus that already existed. I didn't know that the coronavirus was a thing before 2019. Anyway, if mouse hepatitis MHV A59 is IFN resistant, then it will circumvent or penetrate the cell's immune system and infect the cell. Predictions: the MHV will contain an IFN antagonist, and it will circumvent or penetrate the innate immune response of the cells. Methods, they're very complex. If you want, you can pause this and read through them. It'll be easier to explain them once I get to the results. Here we go. Okay, so essentially right here you can see on the right we have two tables. Over here you have the VSV virus, which is known to be sensitive to type 1 IFN, and the MHV A59, which is being tested to see how sensitive it is. Over here for the VSV, you can see this M is kind of like your negative control. There's no virus. This is your virus with just 1 unit of interferon. You see there's a lot, didn't really affect it, but by adding just 10, you can see it was reduced significantly. 100, it was reduced almost to 0. On the other hand, with your MHV A59, start with 1, not affected, 10, 100, 1,000, not really affected till you get to 10,000. So you see the MHV is pretty resistant to the IFN. Discussion. Essentially, the presentation clearly demonstrates that MHV A59 circumvents or penetrates the effects of type 1 IFN. Demonstrates that MHV A59 circumvents the effects of type 1 IFN. That's about it. Thanks for tuning in.